I'm going to pause. Hello and Happy New Year, everyone. My name is Sumaira. I am a patient living with seronegative neuromyelitis optica. I'm also the founder and executive director of the Sumaira Foundation, also known as TSF. We are a global nonprofit organization dedicated to raising awareness of both NMOSD and MOGAD. We are very passionate about building communities of support for patients and their caregivers. We fund research and, of course, are trying to do more in the patient advocacy space. It is my absolute pleasure to welcome you all to TSF's first webinar of the year. Happy Friday. It's the first Friday of the year. We're so excited. So today's programming is all things MOGAD. So we are going to, I will introduce you all to Dr. Levy in just a minute. He doesn't need an introduction, but the focus of today's program will really be about understanding what are clinical trials and how patients can really make a difference in drug development and getting therapies across the finish line and enabling access to therapies, as well as understanding the particular clinical trials for MOGAD that are active at the moment. So I'd like to first thank our sponsor, UCB, for supporting this effort. We are very grateful for the partnership. And just a couple of housekeeping items. As you know, this webinar is going to be recorded and it will be available for replay on TSF's YouTube and Facebook video libraries and soon on our website on our multimedia page. The first half of the webinar will really be about the actual content. Dr. Levy and I will be in discussion about all the different topics, but the second half is really for you to get involved and ask questions. So we're going to do rapid fire questions. You can use the chat function or the Q&A functions to ask Dr. Levy questions. We will get to as many of them as possible. So please don't be shy. Type them in the chat box and we'll get to as many as we can. And so without further ado, it is my pleasure to introduce you all to Dr. Michael Levy, who is a world-renowned neuroimmunologist. He is the chair of TSF's Medical Advisory Board, and I'd like to think he's my friend.
I am. Thank you, Sumaira, for the kind introduction. Hi, everyone. I'd like to start off by just mentioning that MOG antibody disease is really a new thing. It's a new diagnosis that's only been recently recognized. That before this, MOG patients were pretty much lumped into either seronegative NMO, into this sort of wasteland of seronegative NMO that we couldn't characterize further, or MS, multiple sclerosis. And there was really nothing specific for MOG, and we didn't even recognize it as its own thing. And after the NMO world was, after we had 3 clinical trials for the aquaporin 4 NMO world, I think the MOG people were really jealous. They were excluded from trials, from many of the trials, and they really felt like they weren't part of the family because everything was so specific to aquaporin 4. And then we have the MOG antibody test, and it created a new family. It really separated them out from both MS and NMO. It's created a new world for MOG and has captured the attention of the research world and of clinical trial world and of industry. And now we have diagnostic criteria that are coming out in January, and we have a great antibody test, and we have 2 great clinical trials. And it's really, from just a few years ago, created this whole new world just for MOG. And I think for those who test double seronegative, negative for aquaporin 4, negative for MOG, yep, I know how you feel, because it's where MOG was just a few years ago. You feel excluded. You feel like this doesn't necessarily pertain to you. But as new blood tests become available and we start recognizing the differences among the different diseases that kind of all look the same at first, but immunologically are different, your time will come. There will be a blood test for you. But right now we're celebrating MOG, and we're really developing MOG. And we have these new trials for MOG that I'm happy to discuss today.
Awesome. What an introduction. So if it's okay with you, I'd love to ask you a few basic questions about clinical trials, because I do feel that our community, because clinical trials are kind of new for us, we don't really fully understand them as patients. And unless you work in medicine or you work in the research world, how can you possibly know about them? So, can you share with our audience, just in layperson's terms, what is a clinical trial?
Yeah, a clinical trial is a scientific study of a medication in the specific disease. It allows us to give patients accurate information. So when patients come to my clinic and they ask me a question, they say, well, am I more likely to relapse if I use this medication or that medication? If I don't have a clinical trial, I can give you my experience, I can give you my best guess. But if we have a clinical trial, I can give you scientific evidence and say, well, here's what the science tells us. And that's where we get all of our information from. Clinical studies, which are different from trials, clinical studies is where we apply some scientific knowledge, some scientific rationale, or the hypothesis-driven process to data that we can collect from patients, but it's not as good as what's called a blinded placebo-controlled trial where all the biases are controlled for. So if I don't know what medication you're getting, and if I don't know what medication everybody else is getting, then it's the purest form of scientific data. And then at the end of the study, then we can unblind everything and make true conclusions. So that's what a clinical study is. It's where a clinical trial is where you participate in a scientific endeavor to understand the truth about a drug.
So it's safe to then assume that without the patient participation, these trials do not happen, right?
That's right. It's always difficult to recruit patients. Some trials are easier to recruit for than others. Some drugs are difficult to administer. Some have more side effects. Some have more visits to a trial site, so more appointments. Some have MRIs. Some people don't like lumbar punctures, which are required for certain studies. So there's always a reason not to participate in a trial. I think people who ultimately participate want to give some of their effort to the science because it's always a hassle. There's always a hassle involved in participating. And I think it's about giving back to the community. We certainly don't want anybody to sacrifice themselves, their spinal cords or their optic nerves or anything else. We really want to create a trial that is also in your best interest, but ultimately, I think patients are giving back more than they're getting from the trial. It's really about giving to the next generation of MOG patients.
Wow, that sounds really inspiring, actually. It sounds like, you're doing something that's kind of bigger than just you. You're contributing to science, you're contributing to the advancements of therapies and getting things, available for other patients, right? Is that safe
Yeah,
to? Okay.
it's true. I've talked a lot of patients out of it. Patients who want to participate, and I'll say, you know what, though, you haven't tried this other great drug that we think works. We don't have scientific evidence for it, but we think that I personally think it's in your best interest to do this other drug. And then some patients will take me up on that. And some patients will say, you know what, I get it. I know that that's in my best interest, but I want to do what's in the best interest of the community. I want to participate in the science. And I say, great. I just want to make sure, that you know that you have other options that are in your best interest. And if you choose to do what's in the best interest of the community, I love that too. I'm not gonna turn that down.
Right. So outside of the obvious, which is participating in a clinical trial to advance science and medicine and therapies and whatnot, what are some other maybe tangible, intangible benefits for patient participation and like really to reinforce why it's so important for patients to be
Yeah.
involved?
Well, I think for some people, it does offer the opportunity to use a drug that we think is going to be better than anything else that's out there. So
Mhm.
it's access to the drug, it's access to the study team. People who were in our trial, they get to see me, they get to see our coordinators, they get to
Mhm.
be involved, they hear all the news, they're in the family. I think there's a social element to that. And all costs are covered. I don't know if that shouldn't make a big influence. In fact, our institutional review board for the hospital does not want financial pressure to
Mhm.
guide your decision. but everything is free. The drug is free, the travel is free, the bagels are free, everything is free. So that makes
Bagels.
things
Can't say no
easier.
to bagels. That's
Just makes things
awesome.
easier and is the intent.
Okay, that's wonderful. And we are going to get into the particular trials momentarily, but just an overview, like what does that clinical trial journey look like for the patient? So you get enrolled, and then what happens?
Well, first you reach out to the clinical trial site and you indicate your interest. And what we'll do then is review your record to see if you're eligible, because there are a bunch of eligibility criteria. Not
Hmm.
everyone is eligible. Even if you really want to participate, you might not be quite the right type of MOG, or
Hmm.
maybe you don't have the antibody anymore, maybe you didn't relapse recently. So there are all kinds of criteria that will apply. And then if you do meet all of the criteria, we'll invite you in for a screening visit. It might be 2 visits, 3 visits. We get all the blood tests, we get all the MRIs, we get all the eye scans, everything that we need. And then if you qualify at the end of that, then you start the process. And there are different trials. There's one that requires a weekly visit. There's one that requires a monthly visit. There's one that we're planning in the future that will be entirely remote, so no visits. So it just depends on the specific clinical trial.
Okay, got it. And then something that I personally feel very passionate about is the whole diversity aspect of clinical trials. From my very basic understanding, we don't have enough diversity in trials, and we need more of that so that we can accurately represent the community. Any thoughts or ideas about how to get more people involved from all different types of or walks of life?
Well, this is an issue across all clinical trials in
Hmm.
cardiovascular disease and neurology and in every field. Um, it was especially true in aquaporin 4 NMO,
Hmm.
where African Americans made up about 40 to 50% of the patient population. But in the clinical trial population, they were between 0 and 11%. So patients would come in, I'd say, yeah, we have 3 great drugs approved for aquaporin 4 NMO. And if they're African American, they'd say, well, does that one work in African Americans? And I'd say, I don't know, because the trials didn't enroll African Americans. And there are many, many reasons why. We've had seminars and lots of people try to come in and help us figure out why we can't improve our diversity in clinical trials. And there's so many different reasons for that. And most of them are blinded. Most of them are because I don't check to see what race you are. But I think we need to start doing that. We need to start reaching out and being unblinded about that issue. Instead of being blinded and saying, I'll take everybody, I think we need to make a concerted effort to say, well, we need to include more people of this background and this race, and to make sure that we've covered everything, because there may be some biological differences that we have to account
Of
for.
course, absolutely. Wow, that's super helpful. Um, and before we get to the actual clinical trials for MOGAD, for our audience who doesn't know what MOGAD is, what is MOGAD?
I should have started with that. So MOGAD, or MOG antibody disease, MOG, and then A is the antibody and D is the disease, is basically where the immune system decides that MOG protein, myelin oligodendrocyte glycoprotein, it is foreign, is bad, is something that it wants to attack. So the body of the immune system in the MOG patient will try to target MOG and try to remove it, just like it was a bacterial protein. But of course, MOG is present in everybody. All humans have MOG. MOG is found only in the central nervous system. So that's the optic nerve, the brain, and the spinal cord. And typically, when you're born, your immune system knows that MOG is self and it's not going to attack itself. But at some point over the course of your lifetime in a MOG patient, the immune system says, you know, this MOG thing, I don't like it. Maybe it resembles a protein that it finds on viruses or bacteria or something. And at some point it decides this MOG protein is not safe for us. We're going to attack. And then it attacks. And then in about half of people, the immune system goes, oh, shoot, I shouldn't have done that. I apologize. I'm not going to attack anymore. And then it goes away and the antibody can go away. And then in the other half of people, the immune system says, yeah, this is bad. I'm going to continue to attack every time I see it. And then it attacks over and over and over again. And that's the MOG antibody disease that a lot of people on this call have. So We don't know, again, why the immune system makes this error, but it does. We don't think it's genetic. We don't think that this is passed down in families. We don't even think there's a genetic predisposition to MOG antibody disease. We think it's something environmental, some virus, something that triggers this mistake. But one interesting thing about MOG that we've noticed is that if you keep the disease from doing the relapsing part of it, if you just suppress the immune system long enough, that sometimes the immune system will kind of just say, okay, I won't attack MOG now. And it'll kind of wane. It's like after you get your COVID shots, your COVID boosters, your antibodies go way up. You have this great immunity to MOG, to COVID, and then it wanes over time and you lose the immunity and you can get infected again. That's the kind of thing with MOG. If it might be really, really raging against MOG, and then if you keep it cool for a long time, the immunity to MOG goes away and it decides, okay, MOG is going to be okay. So we've seen that a lot with MOG. That never happens with aquaporin 4 NMO.
Wow.
That never happens with multiple sclerosis. Those two diseases are lifelong. But with MOG, it seems like things can really calm down towards MOG more than in any other disease like that.
And how would you compare the prevalence of MOG to NMO?
Well, it's probably a little bit more common than NMO, not as common as MS. Okay, so in classic Western populations like the US and Europe, MS accounts for 97 to 98 out of all 100 cases of demyelinating disease. And then the other few percentage are divided up between NMO and MOG and other things. NMO classically is about 1 in 100,000. MOG may be twice that, something like that. Okay. The Mayo says they're detecting 3 times as many MOG antibodies as aquaporin 4 antibodies, but there's a catch-up effect because the antibody test is kind of new. People who may have had MS or thought they had MS are testing positive for MOG, so there's sort of like a catch-up. In the end, we'll see how much more prevalent MOG is compared to NMO, but it's not as common as MS.
Okay.
Now, in other countries, like in the Far East, in Japan and Singapore, where classic Western MS is uncommon, you see a lot more MOG and a lot more NMO relative to MS.
It's like the reverse.
It's kind of like the reverse.
Wow. Well, very, very helpful. I think this was a great overview of what I like to say MOGAD. It sounds more exotic what MOGAD is. But why don't we get right to the clinical trials? Tell us what you got.
Sure. I'm going to share my screen here. I just need you to allow me to share my screen.
I guess I can.
I just have a few slides on the 2 trials that have already started. Okay. All right. So the first trial is called COSMOG. It's with a drug called rozanolixizumab. That's how you pronounce it, but most people just call it Rosamab because rozanolixizumab is a mouthful. And these slides are from them. And so these are not my colors, but this is from them. And you could see that they want to enroll 104 people across 65 or 70 sites. So that's basically 1 or 2 people out of every site. And it's double blinded, meaning I don't know what you're getting and you don't know what you're getting. Only the pharmacist, who's never going to talk to me, knows what you're getting. And after the period, there's a experimental period that's a flexible length. Then after that, if you relapse or if you withdraw or if you reach the end of the study, then you go into an open label period. And at that point, we know what you're getting. You're definitely getting the drug. You know that you're getting it. I know that you're getting it. Everybody knows you're getting it. That's called the open label period. The study will conclude when there are 58 relapses. That might happen in the treatment arm. It might happen in the placebo arm. We think it'll happen more often in the placebo arm. But once there are 58 relapses, the trial will end. Everybody will roll over into the open label period, and then we will analyze the results. We haven't had that many relapses yet. I think the readout statistically is expected sometime in 2025. So we have a long way to go. This is the basic trial design. The print is a little small. I apologize, especially for MOG people after optic neuritis. This is hard to read. The point is that there's a 50-50 chance of being in the placebo arm. And the placebo arm is not allowed to use any other immune suppressive medication. You can't be on IVIG, you can't be on azathioprine, you can't be on prednisone. You have to be on placebo. Now, if you have aquaporin 4 NMO, you know the risk of being in the placebo arm is very severe. Because if you have an optic neuritis with aquaporin 4 NMO, you might lose vision, and that's a big, big deal. But with MOG, the consensus of the experts is that if you catch it quickly and you treat it aggressively, that MOG relapses are not as damaging and not as severe, not as lifelong, that they heal better. And so the placebo ethics is allowed with MOG, whereas with aquaporin 4 NMO is a little bit of a gray zone. So that's why the placebo arm here has no background treatment allowed. The inclusion criteria are that you have to be an adult. We thought about trying to enroll kids. It was really difficult to get that through the regulatory agencies. You have to be an adult. You have to have a MOG antibody, and you have to have had an attack in the past year. And why is that? Well, if you hadn't had an attack in the past year, the worry is that you're going to get over it. And so if you don't relapse, maybe it's because you're getting over it, not because of the drug. So if you've had an attack in the past year, we think your disease is more active. So that's why that criteria is there. Now, there are a lot of things to do in the study. Here's a schedule of events just to show you that every column here is a visit. So at week 1 and week 2 and week 3 and week 4, you can see all the X's required. So for example, we have to review your medications every single time. We have to see if you have suicidal ideations. We have to see, we have to do an EKG. We have to do all these different things, physical exam, neurological exam. There are many, many things. Each visit probably takes 3 or 4 hours. You're there with the study coordinator and with the nurses and with everybody else, the whole study team. And it is a burden. It's a long visit for each time that you do it. But after a certain period of time, I can't remember how many weeks, then you can start to do the treatments at home. So a nurse will come to your house, help you do the infusion, and then you're done. And then you still have to visit our site every 3 months or so. Now, what are the readouts? Well, we're looking to see if you relapse or not. If you relapse, then the drug failed. And if you didn't relapse, then hopefully the drug is accounting for that. And we look for relapses by MRI, by ophthalmological exam, by clinical exam. So if you think you're relapsing, then we'll do a whole workup to see if you truly are. And then if you truly are, then we have an adjudication process to confirm that. And if you didn't relapse according to the criteria, then you could get to stay in the study. If you did relapse, you move to the open label arm. And we don't tell you what you were on at the end of that relapse. So you don't know if you got the drug or if you got the placebo until the end of the study. So everybody has to be done, and then you get to know. So we just launched this study. We enrolled our first 2 patients, and one of them has moved to Arizona, and one of them is now taking the treatments at home. And I think, it's been going well. Certainly, it's hard to get everything started. These are weekly infusions under the skin. I should also mention this process. The volume of drug is not very much, but it's about 4 mLs, or just under a tablespoon. And the FDA says you can't inject a tablespoon of medication in one shot. You have to do an infusion. So we put this tiny volume into this little pump, and it goes through this little tubing into a patch in your skin with a tiny needle, and then it infuses over 15 minutes. So that's how this drug gets under your skin. And you don't really see it. After you take the patch off, it's kind of gone already. And it is weekly. And the hope is that if it's approved, FDA approved, that this treatment would be available at home with this little pump. You can travel with it in theory. And that's how this drug works. So, Sumaira, I do have information about other trials that have launched as well. Should we take questions about this one first or just touch on the other trials?
I'd say let's touch on the other trials and then open it up for
And
Q&A.
then open it up.
Yeah.
Fair enough. So there's a second trial that has launched. This one is, don't pay any attention to this icon down here. This one is launched by Genentech. It's called Meteoroid, and it uses satralizumab. So for the aquaportin 4 people in the audience, you know satralizumab is already approved for your disease for the aquaportin 4 version of NMO, and now we're testing it in MOG. This drug blocks the interleukin-6 receptor, which is involved in a lot of inflammatory diseases of the central nervous system, and we think it would be helpful in MOG. We have some case reports to support that. And the trial design is fairly similar. 50-50 chance you're enrolled into either a satralizumab injection or placebo. The big difference is that in the placebo arm, you are allowed to stay on your IST, which stands for immunosuppressive therapy. So you could stay on your Cellcept, azathioprine, or your low-dose prednisone while you're in the study. Whereas with the COSMOG study, you cannot. But it's still a 50-50 chance, and you also have to have a relapse in the past year, and you also have to have the MOG antibody. So otherwise, they're fairly similar trial designs. They have to enroll a few more people than the COSMOG study, 152. And, and the one other inclusion criteria that makes it a little bit easier for this study is that you don't have to have an attack in the past year. You can have had 2 attacks in the past 2 years. So that opens it up a little bit more. This is an injection. The volume is 1 ml. So that is allowed to be injected all at once. So you get one injection at a time every month, not every week, but every month. This study is also looking to prevent relapses. It has basically the same endpoint to prevent relapses, and how we adjudicate that is basically the same. And then the last trial that is going to launch eventually is a pill called tolebrutinib. This one is being developed by Sanofi in multiple sclerosis. They've had really good success in multiple sclerosis. Here's an example for MRI lesions showing in the 60 milligram group that people who use tolebrutinib have many fewer MS lesions. There's a lot of biological rationale to use it in MOG, and the class of drug called BTK inhibitor is being developed in MS. Many other companies are doing it. It's either many different types of BTK inhibitors. Tolebrutinib is just one of them. There are many trials going on in MS right now. This is the only one going on in MOG. Now, the design is different. This is a phase 2 study. There is no placebo arm. Everyone gets the drug. We're only enrolling 25 people. They're only going to be on the drug for 1 year. We do lots of blood testing for biomarkers and safety as well. And it's entirely remote. So you never have to show up in Boston. So you do everything through Zoom. All your blood tests are done locally. And if you relapse, God forbid, we need the help of your local neurologist for treatment. That one, expected to launch this month, probably won't happen. Probably going to take a few more months to get out there. And there are lots of future preventive therapies, Sumaira, that one day we can talk about that are on the research experimental level.
Awesome. All right. As always, as expected, that was fantastic. So I think now maybe we just open it up to the audience. There are a few questions that have already come in, so I'll read them to you and then you want to answer. Does that work?
Sure.
Okay, great. Somebody says, please tell us the results of rozanolixizumab.
The rozanolixizumab trial, that's COSMOG. There are no results yet. They've only enrolled, I think, 10 out of the 100 so far. It literally just launched just in 2022, so we don't have results yet.
Okay. When can people expect results?
I think we do expect results around 2025. It depends on when we get those 58 relapses. Then we'll
Got
have
it.
all the data.
And actually, I have a very quick question about that. Are the sites only in the US right now?
No, no. In fact, oh, I have a map of the world.
Ooh, show it.
I have to dig it up.
Okay.
Maybe we'll post it on your website.
Would love that. I think it'd be
Um,
good for our international audience to see
yeah,
where they could enroll in studies.
many different countries, and still growing. A few new sites, but I think there are about 60 sites already activated or to be activated. About 8 in the US, I believe, maybe 10, 8 to 10, and then many sites in Europe and Asia and everywhere else.
And do you suspect that with time there will be more and more sites added?
Maybe. We're trying to concentrate at population
Mm,
centers, so we can't make it available everywhere.
okay.
Um, but we want to try to capture as many population areas as we can.
Okay, great. All right, so we have a question. Can we expect a cure from rosanolixizumab? How is it working?
So the mechanism, I didn't touch on this,
Mm,
of rosanolixizumab is really different. We've never had a drug like this. There's never been one approved before, but this is a new mechanism to force the recycling of your own antibodies. So MOG antibodies and other antibodies all get recycled and destroyed with this drug. And it's given once a week. So as soon as you get the drug, then all your antibodies go away and your bone marrow is forced to make all new antibodies. And when your bone marrow remakes all your antibodies, it doesn't tend to focus on the bad ones like MOG, which developed somehow from some environmental exposure. It tends to produce all your good antibodies, like to things that you were vaccinated against, MMR and all those vaccines you got as a kid. All those antibodies come back, and over time, when you keep doing this every week, forcing your bone marrow to make all new antibodies, you basically lose your MOG antibody. Now, it might come back after you stop the drug. We don't know. That's something that we are looking at. But if you think about it, it's kind of like plasma exchange. When you get plasma exchange, you get your blood taken out, it's spun down, all your antibodies are thrown away, and your blood cells are returned. So it's like getting plasma exchange every week.
oh,
Now, this
wow.
mechanism has been tested in many diseases where plasma exchange and IVIG are helpful, like myasthenia gravis and CIDP. And in those diseases, a lot of these drugs are already approved because we know. So that's how we know that they work.
Wow, cool. All right. How do you determine eligibility and when would you remove a patient from a trial?
Oh, so eligibility is determined based on a whole set of criteria. It's like 3 pages of criteria. And we just go through one at a time. Are you over age 18? Check. Are you under age 89? Check. And we just keep going down the list. Did you have a MOG antibody test? When was your last relapse? What kind of relapse was it? How was it treated? What medication are you on now? What other diseases do you have? Do you have cancer? All these exclusion and inclusion criteria. And then at the end of it, hopefully you qualify and then you can enroll. And when would a patient get kicked out? It's pretty rare that a patient would get kicked out. Was that the question? Getting kicked out?
Yeah, when would you remove a patient from a trial?
I mean, if they didn't show up multiple times, if they developed cancer and there's something dangerous about the drug, if they're suicidal, it'd be pretty rare that we would remove a patient from the trial.
And that actually just triggered a thought for me. You mentioned that there are 8 to 10 sites in the US for
For
this
Cosmo.
particular trial, right? So let's say there's a patient in a state where a site is not available. Could they come to someone like you? Like, do you accept out of
Yes.
state?
Yes, we encourage that. Our
Okay.
first 2 patients were out of state. They were not in Massachusetts.
Okay.
One was from California and flew every week. Thank you, JetBlue.
Mhm.
And the other was from Albany, or Rochester, New York,
Okay.
and would fly in as well every
Okay.
week. And
Wow.
it's difficult. That's why it's a real commitment. But there are no sites in Rochester, New York. I think Boston is the closest one to Rochester, New York. So thank you, JetBlue from Jim Bradman.
And
So yeah, and all the travel is covered, of course, because we want to try to make it as easy as possible for patients without burdening them.
totally. And in terms of the eligibility, is there a screener available online that's public for people who are interested in joining? They could take a look in advance.
Yeah, the criteria are certainly posted online, but it's not the easiest thing to go through on your own.
Okay.
Try on clinicaltrials.gov, the inclusion criteria are posted there. And I believe each trial has their own website that kind of helps you go through to see if you'd be eligible. But there are always a few lingering questions, and it's really helpful to just reach out to us.
Got it. How do you define a relapse? Is this limited to intensity of the attack or location with regards to the central nervous system?
So there's two different definitions for attack. One is attack that occurs before you enroll in the study, and one is an attack that occurs after you enroll in the study. The definition of an attack after you enroll in the study is very, very, very specific. The FDA wants every attack to be supported by MRI evidence and clinical evidence and optical coherence tomography if it's an optic nerve lesion, and many, many other points of evidence to support the drug company saying that their drug works or doesn't work. But before you enroll in a study, sometimes your doctor may not be 100% sure. They may not have gotten the MRI in time. They may not have seen exam findings that support a relapse or not. So it's more of a judgment call before you enroll in the trial. Generally, what I try to do is I try to look for clinical worsening plus an exam finding change. So if you say that there's a problem with your left eye, blurriness, for example, then I want to see that you can't see the vision chart as well. And then on top of that, then I want to see that there's an MRI lesion. So that's a little bit of an extra level of objective evidence to support a relapse. That's how I do it in my clinic.
Okay. Here's a very interesting question, or a comment rather. I had looked into the trials. It didn't appear to be available for patients in California. I'm currently taking Hizentra weekly. I have not relapsed. If I stop treatment or it gets delayed, I'm decline in functioning and more pain. I have MOGAD and my cousin has NMO.
Ooh.
Ooh.
So we don't see a lot of MOG in families. And I've never thought about MOG and aquaporin 4 in the same family. That might be a first. So I would
Wow.
love to talk to you more about that, try to understand how that happened, and if it's a
Yes.
blood relative or not a blood relative with a common exposure, for example. So I'd love to hear more about that. So no California sites. That's not good. We really should have a California site. I believe there's one in Northern California.
Is it Stanford?
I think so. Yeah, Stanford is a site. Yeah, but
Yeah.
not Southern California. We don't have a site in Southern California.
Okay. Well, to this person, please email us so we can get you connected to Dr. Levy. But sounds like a very interesting case of having MOGAD and NMO in one family.
Yeah.
All right. Next set of questions. If you have a patient in one clinical trial and another trial starts that you believe would be of greater benefit, will you switch them to the new trial? And can you tell us in layperson's terms how to explain how Rosanna Ligzixim, I still can't say it. Rosie, how does Rosie work? Rosie.
All right. So in a clinical trial, if you are enrolled, it is my obligation to always keep you updated about treatments that might be in your best interest. So let's say, for example, you're in a clinical trial and a new drug is approved for MOG. I have to tell you about it. And I have to tell you whether or not it would be in your best interest, because the Institutional Review Board doesn't want to sort of trap you into participating in a trial. They always want it to be in your best interest with your full consent. What was the second part of that?
How does one explain how Rosie works to their
How
friends?
rozimab works, right? So the way rosanolixizumab works is kind of like an internal filter. It just washes out your antibodies and recycles them, and then you make all new ones, kind of like a reboot of all your antibodies.
Okay. Why do MOG patients experience seizures, and can we say uncontrollable seizures around 20 episodes is a relapse?
So, okay, great question. So MOG is also expressed in the brain, and we do see seizures, especially in kids and in teenagers. So that's a common thing with MOG that we don't see with aquaporin 4 NMO. And here's the weird thing about MOG. MOG can cause seizures in two ways. It can either cause inflammation, and the inflammation itself is triggering a relapse. And you can see that on the MRI. In that case, you want to put out the inflammation, and hopefully the seizures will die down. The other way that MOG causes seizures is it causes damage in the brain, and then that damage causes a scar, and the scar causes the seizure. And so if you don't have any inflammation in the brain, and the seizure is being driven by the scar, then steroids and other anti-inflammatories won't help in that situation. So you really have to figure out what's triggering the seizures. We have seizure drugs that are good to prevent seizures from scars, for example, but that would be different treatment altogether.
All right. Okay. Oh, we have a question from Mr. Michael Devlin. The trial sites are all at top academic medical centers. It seems like the average trial participant might benefit from better quality of care than what is available to them locally. Is this a potential benefit of participating in trials?
I think so. I think having access to a MOG expert site is a good thing. You can ask all your questions, get all your questions answered. You can learn from others, get connected to other MOG patients. I do think it's better that way. I've heard from a lot of my MOG patients that their local neurologist only sees 1 or 2 MOG patients and doesn't have a lot of experience. So I think it could be a good thing. Travel is a burden, I have to admit. Even the people who were in our rosuvastatin trial had to fly into Boston Logan Airport every week. It's a hassle, but I do think that having access to a MOG expert site is definitely a benefit in participating in the trials.
Okay. What happens once the trial is over if the drug is successful? That's something we want.
If it's successful, then what'll happen is you get to stay on the drug free of charge or paid for by the pharmaceutical company until it's approved by the FDA and then covered by your insurance. So then you do transition. So hopefully you'll never be off the drug if you want to stay on it.
Okay. While you have mentioned that MOGAD is not genetic, I am curious, given that there was a case of myasthenia gravis in grandparents, how did you come to the conclusion that this has no genetic component?
I wouldn't say I concluded it. I would just say that we don't see evidence for genetics yet. So there's 2 ways that we do that we look for that. One is we look to see whether the prevalence of MOG in families with autoimmunity is more or less than in families without autoimmunity. And with MOG, we have tons and tons and tons of cases where everyone in the family has no autoimmune disease, and there's just one person with MOG. In contrast, with aquaporin 4 NMO, we see exactly the opposite. We have tons of family members with lupus and Sjogren's and rheumatoid arthritis and everything else with aquaporin 4 NMO, but not with MOG. So that's one way, is epidemiological studies. The other way is through genetic studies. And they've only published a few so far, but in the studies that have been done in MOG, there are no genetic common variants. Nothing in people with MOG don't seem to have any genetic mutations that we could find that are common to MOG.
All right. We have another question here. If I was to relapse from an infection, is there a time period to expect the relapse? So I guess how long after an infection would you expect a relapse?
So during an infection, your immune system is activated just globally. If you get COVID or flu or whatever, your immune system is activated and you're going to fight off the infection. And then about a week or 2 or 3 weeks later is when we tend to see that an infection could trigger a MOG relapse. Doesn't usually happen while you're sick, usually happens after you've recovered from the infection. Just, I don't know why, but that's when it happens. And I wouldn't say you're out of the woods until about 4 or 5 weeks later.
Okay, to our live audience, we have time for about 3 more questions, but until we get some more, Dr. Levy, I have a question for you. I have heard that the best time to test for the MOG antibody is during a relapse. Can you explain why and like how the titers and all of that work?
That's observational, meaning we haven't proven that definitively, but it seems like that the MOG antibody levels are highest at the first event of the disease. So like at your first optic neuritis, and then it kind of goes down after a while. So if you have an optic neuritis and you get steroids and plasma exchange, and then 3 months later, somebody says, hey, we should have checked for MOG antibodies. Let's check them now. They might be gone, or they might be really low, and you missed your chance to see the really high levels. We don't tend to see that with aquaporin 4. Aquaporin 4 levels just tend to stay up.
Right.
But with MOG, they do tend to decline. So you kind of want to capture them as soon as you can. Now, other things besides plasma exchange can reduce levels a little bit. Steroids can a little bit. Other drugs can. Plasma exchange, of course, removes it. Rozanolixizumab, of course, removes it. IVIG also dilutes MOG antibodies. So all these different medications can make it harder to detect.
It's kind of fascinating that that's how it works. Are there any other antibodies like outside of neuro that are kind of like that?
Yeah, so there are a lot of antibodies for different rheumatologic conditions that can come and go as well. And sometimes you just have to be lucky to capture it, or sometimes the best time to capture it is when you're sickest.
And for those patients who are double seronegative like myself, how often would you recommend getting tested for retested for antibodies?
When you're sick, and
Okay.
then outside of that, probably every 6 months, just by random chance, we might capture it then. We've seen people who live at low levels. They might test positive at like 1 to 20 and then go negative and then positive again, negative again, depending on what medication they're using, or even people who are not using medication, their antibody levels still fluctuate. Some people's antibody levels don't really fluctuate. I had one lady who wanted to test every month. So I gave her a script and insurance covered it. And we were testing it monthly. And she'd be at 1 to 100, 1 to 100, 1 to 100, and then 1 to 40. Hey, that's good. But then back up to 1 to 100. And then one time it went up to 1 to 1,000. She got all worried that there was no attack, and then back to 1 to 100. So there's some variability in the antibody levels that's occurring in the background outside of any disease activity that we need to understand better.
That's fascinating.
Mhm.
Okay, we have 3 questions left. So, how long after becoming MOG negative would you trust that the MOG antibodies are gone for good? Can treatments like IVIG cause a false negative?
Yeah, IVIG specifically causes a false negative. It dilutes the MOG antibody with all the antibody you're getting in the drug. And so really after IVIG, I usually wait 8 weeks to be sure that the MOG antibodies are really gone. Even after they're gone, I don't celebrate. I don't pop the champagne cork. I wait. I usually test again in 3 months. And then if it's really, really, really gone, then 3 months after that, it should be gone as well. Now, I had some patients who it was gone, it was gone, it was gone, and then it blipped back up into positive range. And they get all worried. Should I go back on treatment? Should I not? We don't know. And we don't know if that's meaningful. And then we test again, it goes back down. It's really hard to know. The antibody test has only been available for 7 years now, and we don't have all the answers.
Can a particular diet prevent relapse?
Ooh, thanks for plugging that diet study. We don't know,
Yeah.
but we're looking into it. There's a fantastic graduate student from Johns Hopkins named Alexis Martin,
Lexi, yeah.
Lexi, and she's interested in a diet study. So we're launching this diet study to divide patients into 3 arms randomly, either a classic Western diet, a Mediterranean diet, which is like, salmon and things like that. And I think it's a lactose-free diet. Hope I'm not messing that up. And you're in the diet for 4 weeks, and you have specific recipes and things like that. And then we're going to see, what does your quality of life look like? What happens to your MOG antibodies? So hopefully we'll have some answers for you.
Okay, and the last question, I think we answered it a little bit, but maybe we could just repeat some version of the answer. If you have MOGAD and other autoimmune issues, how does a patient identify a relapse of MOG?
Oh, that's a tough one, because
Oh,
let's say you have MOG and arthritis,
oh,
and your arthritis flares. How do you know it's not a MOG issue? It's hard. Typically, I have a low threshold for ordering MRIs. I love MRIs because MOG attacks almost always show up on an MRI if you get the MRI in time. So don't get it too early, don't get it too late. It's just a perfect time for it. And also ophthalmologic exams and optical coherence tomography scans. And what we're looking for is more objective evidence of inflammation. So just because you have more symptoms, it could be from the damage that was done. What we're really looking for is there evidence of new inflammation in the central nervous system? That is really what a relapse is.
well, this was fabulous. Thank you so much, Dr. Levy, for such wonderful information about MOGAD, the clinical trials, the particular clinical trials, and why patient participation is so important. For those who are interested in learning more about the COSMOG study, that's the UCB clinical trial for Rosanna Lixizumab, Rosie, we'll just call it that. Please visit www.cosmogstudy.com. If you have particular questions for Dr. Levy, you can send them to the foundation. So that's info@sumairafoundation.org, and we will forward them to Dr. Levy. But thank you so much. This was so
Pleasure.
fun, as always.
It's been a pleasure.
Bye, guys. Have a good Friday.